Mount Sinai, and I would like to present to you our paper on pathogenic germline variants in 10,389 adult cancers. This paper was published in Cell last year and was a study I conducted while in the Dean Lab at Washington University, along with the Pan-Can-Alice Germline Working Group with many individuals across different institutions involved. In the gist, you can see here the graphical abstract highlight the research pretty well. We studied genetic predisposition in cancer, and here's an overview. So first, I'd like to go into the motivation of this study. We all probably know of unfortunate examples like this in a family where you see multiple family members having cancer. There you can see the grandmother has ovarian cancer, mother has breast cancer, ovarian cancer, and the granddaughter has a breast cancer, all at a relatively early onset age. And so a human geneticist will tell you that there's probably a genetic disposition passing along this family. Eventually, this was found to be a classic BRCA1 pedigree, and we all know of genes like BRCA1, BRCA2 involved in breast ovarian cancer predisposition. But we don't really know that much besides from these key genes and in these selected cancer types. So that gives us a good opportunity to use the data set we have to ask what genes and variant predispose to cancer and how. The enabling data set of our study is the Cancer Genome Atlas, one of the most high-profile cancer sequencing projects in the US to date that has accumulated over 11,000 individuals, really having a series of really important publications in 2005 up until recently that really empower a lot of the largest genomic findings in cancer. And obviously, we have multiple molecular data type that's really investigated in the Pan-Can Atlas. For today's talk, I will focus on the germline part in our paper. So going back to zoom in to the graphical abstract, we characterize germline predisposition eventually in 10,000 cases that pass QC in 32 cancer types. In total, there's more than almost 1.5 billion germline variant calls. And using a series of algorithms, we're able to zoom in to 871 predisposition variants, and these include truncation, missense, and copy number variations. So that gives us one of the first biggest cross-cancer type landscape of the important genes that involved in predisposition of each cancer types. We found that across all these samples, there's around 8% of cancers, adult cancers in TCGA, carry these predisposition factors. And many of those translate to a somatic second hit, which then transcribe to a transcriptional effect, and then some of which we have validated as to have functional consequence. So first, let's go into how we generate a variant set data. So here, if you look into the paper manuscript, when you're reading the manuscript, we first talk about how we generated the data. And the challenge here, and also the data set of sample size and the 33 cancer types involved are shown here in details. So one of the hardest challenges in germline study, especially compared to somatic mutation study in cancer, is that you have a lot of variants. And so you're going up against a really high signal noise to signal ratio. So imagine we generated almost 1.5 billion variant calls using 3 variant callers, GATK, BarScan, and Pindel. And this obviously is empowered by large-scale cloud computing and also Genome VIP, our software that enable us to generate workflows to conduct cloud computing. And then even when we zoom into exomes, we still have almost 30,000 variants per individual. And so obviously most of these are not pathogenic. They're explaining natural variations among us humans. They can explain that why we have different color hairs, different heights, etc. So we then also developed tool called Charger that was published recently in Bioinformatics that allow us to prioritize these variants based on the released AC and G guidelines. So that rolls down to around 30,000 variants across all samples. And then eventually we do further QC and then zoom into the cancer-related variants, eventually driving down to 435 pathogenic variants and 418 likely pathogenic variants. I really like to acknowledge a lot of folks at ISB here that allowed us to do cloud computing, especially Elias Group, as well as my colleagues Jay Mashal, who built GenomeVIP, and Adam Scott, who's a primary developer of Charger, that really enable us to come down to these critical pathogenic variants in the study. And so if you look into the paper, after that, this really allow us to come into one of these large-scale pathogenic variant landscape where you can look across cancer type, which cancer type have the highest percentage of pathogenic variant carrier, and that's obviously pheochromocytoma and paraganglioma, which is associated with Mantoux syndrome that's known to have a lot of pathogenic variants. And also you see ovarian cancer, breast cancer pretty high up there, whereas other potentially more environmental driven cancers have low germline predisposition. We know a lot of these predisposition genes to be in tumor suppressor genes shown in blue, but we also see quite some variants, even outside of PCPG, which is known to have red variants, also have oncogene variants. And so we identified a percentage of carriers in each cancer type, and each of the genes were identified to have specific enrichment in specific cancer types. And so here to zoom in on that, well-known examples like BRCA1, 2 in breast cancer, showing that our approach is really working well. Ovarian cancer has even a higher BRCA1, 2 predisposition. At the same time, we have a lot of other interesting candidates. In kidney cancer, we see VHL, BAP1 at a germline level. At GBA, interestingly, we see potentially BAP1B has enrichment of pathogenic variants. In stomach cancer, on the other hand, we see ATM, POLB2. And so here's a lot of interesting information to digest here, and we encourage readers for the specific cancer type of interest to read more into it. So after having this landscape, we want to see whether this variants are doing additional things. And so one of the main mechanism that the pathogenic variants are known to drive oncogenesis is through Knudson's two-hit hypothesis, which is really first proposed by Nordling in 1950s, but statistically shown by Knudson in 1971. And so imagine germline variants are the variants that you are born with and inherited from your parents. Well, most of the case, it's in the tumor suppressor, like BRCA1. So even if all your cells have one disabled copy of BRCA1, the other copy of BRCA1 will still go around and repair DNA. However, in a rare unfortunate event, one of your cells may acquire additional somatic mutation on the other allele, and this disabled the other copy of BRCA1. And now that cell may be in trouble because that enabled it to rapidly acquire mutations, some of which might be oncogenic and eventually lead to development of a cancer cell. Well, the second hit comes in two flavors. One is so-called loss of heterozygosity, where this outer allele is simply deleted, and thus it would seem like this germline variants become homozygous. The other is so-called biallelic event, which is similar to what is shown here. The other allele simply has the other different somatic mutation. So we went on to discover and characterize these kind of two-hit events, especially taking advantage of the somatic genomics data available in TCGA. And we see genes that have a lot of these percentage variants going through loss of heterozygosity, especially tumor suppressors, so well-known BRCA1, 2, but also other genes like BAP1, SDHB, VHL, and maybe VANCAM and F1 all have high degrees of loss of heterozygosity, really validating these pathogenic variants are undergoing these additional somatic events, potentially driving tumorigenesis. And the other type of event, as we mentioned, is a biologic event where you can see here shown in red, each germline variant is linked in one sample to the other somatic mutation affecting the same gene. So for example, this sample here in the gray line has a nonsense variants here at germline level, and also has a friendship somatic mutation, disabling potentially the two copies of their ATM. So we observed these two hit events. So next, what will we do? Well, another thing that we would look into is the somatic expression profile. Say if you have a truncation in tumor suppressor gene, would you actually see that correlate with actual lower expression of the gene? And so here we conduct a formal association analysis. And just to zoom in on some of the results, you see that as predicted, a lot of the truncations we see in tumor suppressors do correspond to lower expression of those tumor suppressor genes. Interesting phenomenon that really pop up here is for the pathogenic variants we're identifying in the oncogenes are often associated with higher expression of the gene in the predisposition carrier. And this has previously been shown for somatic mutations that the tumor tend to favor these activating allele, so that when they have these potential activating allele in oncogenes, they find ways to upregulate it, and so you then see upregulated level of the gene expression. So that really is an interesting phenomenon. Here, I like to especially point out red. And so here, I'm not going to go into this copy number variation analysis. We put a lot of effort into this, discovering the CMV. Ego, previously a student at Leadings Lab, did that. We also look into other type of supporting pathogenicity, pediatric cancer, overlapping with somatic mutations, overlapping with post-translational modification sites. But I want to jump into what we found of these red variants that are interesting because they correlate with higher gene expression. We really assume that they might have some kind of functionality. And so we conducted functional analysis of the red variants. Besides from the known pathogenic ones, we also prioritize some variant of previous unknown significance that we characterized in this paper. So here, you can see that here's a result of that characterization. Clara, a previous tech in the lab, conducted this analysis. So here, for example, are red the germline variants that we're interested in characterizing. The somatic mutations obviously are more likely to be pathogenic, and there's also other co-localizing germline variants. And so we want to see that if they show a gain of function, and so that corresponds to phosphorylated downstream MAPK compared to wild type under activated state, or not non-activated by BDNF state. And so you see that this non-pathogenic variants definitely have that upregulation. This 918 and 912 also has that upregulation. The other variant that we show to be found in these samples and also clustering seems to have a slightly elevated sample activity. However, we don't have enough data to really prove that it's shown that gaining of activity. Another phenomenon here is that in vitro, we didn't really see that obviously higher regulation of the protein, and potentially there are more contexts that are required to really activate these germline variants that require further investigation. And so in sum, I hope this paper presents a comprehensive view of what genetic predisposition, especially for intermediate and high-risk allele in predisposition genes, are across a wide array of cancer types. We show that these predisposition variants are located in different genes, but sometimes they're shared across genes in different cancer types that can potentially aid the genetic screening for those other cancer types besides from the well-known breast, ovarian, prostate cancer that have genetic predisposition, for example. We also showed evidence of somatic second hit events, expression effect, and potential functional consequence that really provides tools and possible ways for investigators to, in the future, further characterize these germline predisposition and how they may actually affect oncogenesis in the downstream route. And this study, we have a lot of collaborators to thank. You'll see that in a full list of co-authors. We hope that you enjoy reading this manuscript. The data of the germline variant calls, as well as the pathogenic germline variant calls, if they have the sample ID, is a protected data that requires TCGA-DB get access. And if your group have that access, you can assess them in GDC, and I will post that link. And thank you all for your attention.